Good afternoon. Today we are going to discuss about action potential in the cardiac muscle. We will briefly revise about action potential in the neuron. In the neuron, the resting membrane potential is minus 70 millivolt. Now, when you apply a threshold current, minus 55 is the firing level. Then there is influx of sodium into the cell, which causes depolarization of the membrane. But the sodium channels, they are fast to open and fast to close, and the closing of sodium channel or inactivation of sodium channel is voltage and time dependent. So there is a overshoot, but it doesn't reach the equilibrium potential for sodium ions. Sodium channels become inactivated and potassium conductance of the membrane is there. So potassium moves out of the cell, which is responsible for the repolarization phase. Towards the end of repolarization, there is still potassium conductance of the membrane. So the membrane hyperpolarizes a bit, followed by bringing back to the resting membrane potential. Till 1/3 of the repolarization, the sodium channels are inactivated. So however the strength of current you increase, you cannot generate another action potential. This is the absolute refractory period. Now, in the cardiac muscles, we have discussed there are two types of action potential. One is the fast response. The next one is the slow response. The first response action potential occurs in normal atrial and ventricular myocytes and in specialized conducting fibers, while the slow response in the SA node, AV node. Now we will discuss about the different phases of this action potential. So a threshold stimulus, what does it cause? It causes opening of the sodium channels. And there you can see there is a rapid rise in the membrane voltage in phase 1. This is phase 0, sorry. This is due to opening of sodium channel. The rise is steep out here. This is due to opening of sodium channel. The membrane conductance for sodium increases and there is a small amount of overshoot. Then the sodium channels, they become inactivated. Once the sodium channel becomes inactivated, however the strength of current you increase, you cannot generate another action potential. This is the effective refractory period. Now we come to the phase 1, that is genesis of early repolarization. The brief repolarization results in the notch between the end of the upstroke and the beginning of the plateau. Repolarization is brief because of activation of transient outward current, RTO, carried mainly by potassium ions. The cell is briefly and partially repolarized as a result of this transient efflux of potassium. This is due to transient efflux of potassium. You see in this picture, the action potential and the contractile response in a cardiac muscle begin nearly at the same time because of presence of gap junctions. So this is a comparison between cardiac and skeletal muscle. You can see in the cardiac muscle, the contractile response and the electrical response, they nearly begin at the same time. But what happens in a skeletal muscle? In a skeletal muscle, it begins 2 milliseconds after the electrical response has occurred because release of neurotransmitter from the presynaptic part to the synaptic cleft acting on the postsynaptic membrane on the receptors then traveling along the T tubules, followed by excitation, contraction, coupling. All these require a time. So there is a gap of 2 milliseconds because there are no gap junctions present here. Now see this picture. Here you can see the principal ionic currents and channels that generate the various phases of action potential in the cardiac cells. Now phase 0, we have already told, this is due to opening of the fast sodium channel, this upstroke. Phase 1, the chemical and electrostatic forces both favor the efflux of potassium through ITO channels, and there is a partial repolarization that occurs. Now phase 2, this is the Plateau phase. There is net influx of calcium through calcium channels, which is balanced by efflux of potassium through IK, IK1 and ITO channels. So they balance, the calcium influx and the potassium efflux balance one another. As a result, you see there is a Plateau formation. Now we come to phase 3, the chemical forces that favor the efflux of potassium through IK and IK1 channels predominate in this part over the electrostatic forces that favor the influx of potassium through the same channel. So in this instance, efflux of potassium is more as compared to the influx. Now we come to phase 4, the chemical forces that favor the efflux of potassium through IK and IK1 channels very slightly exceed the electrostatic forces that favor the influx of potassium through the same channels. So these are about the chemical and the electrostatic forces, this diagram. Now you can see the action potentials in different parts of the cardiac muscle. The A is action potential recorded in the epicardial cell. B is in the mid-myocardial cell. While in the endocardial cell, you can see it is the C. The notch you can see it is different. The shape also is different. Why is it like that? We will see. There is difference in distribution of channels, ion channels in different parts. And this is the reason you can see the shape size vary from different parts of the cardiac muscles. Now, we have already discussed about the genesis of the early repolarization due to transient outward potassium current by ITO. Now we will come to phase 2, that is the genesis of the Plateau. So during the action potential Plateau phase, calcium enters the myocytes through calcium channels. So there is entry of calcium into the cell. During the flat portion of phase 2, this influx of calcium is counterbalanced by the efflux of potassium. So there is, in the Plateau, there is influx of calcium and there is efflux of potassium ions. They counterbalance one another. So the phase becomes a Plateau. So the phase becomes a Plateau. Potassium exit occurs through channels that conduct mainly ITO, IK and IK1 currents. We have already told that the ITO is responsible for phase 1. The IK and IK1 currents, we will see them again later. Calcium enters the cell via voltage-gated calcium channels. Two types of calcium channels are there. One is L type, another is T type. L type is long lasting. The name is L because they are long lasting. They are the predominant type of calcium channels in the heart, and they are activated during the action potential upstroke. When the membrane voltage reaches about minus 20 millivolt, minus 20 millivolt. This L type of calcium channels are blocked by calcium channel antagonist. Example of them are verapamil, amlodipine, diltiazem. Now there is another type of calcium channel, that is T type or transient type. These are much less abundant in the heart. They are activated at much negative voltage, that is minus 70 millivolt, while the L-type calcium channels, we have seen they are activated at minus 20 millivolt. These channels, the T-type of calcium channels, they also inactivate quickly as compared to the L-type of calcium channels. L-type calcium channels are more abundant. Opening of calcium channel results in an increase in the calcium conductance, and so the action potential upstroke also increases. In the phase 2, we have discussed that the calcium influx balances the potassium exit, and this is responsible for the generation of the plateau. The calcium influx during the plateau is involved in the excitation, contraction, coupling in the cardiac muscle. During the plateau of the action potential, that is the phase 2, the concentration gradient of potassium across the cell membrane is virtually the same as it is during the phase 4. So, the concentration gradient for potassium in phase 2 and phase 4 are nearly similar. Now we will see the different types of potassium currents. There is some current which is called inward rectification. Inward rectification is a characteristic of several potassium currents, including the IK1 current. For these channels, large potassium currents flow at negative values of Vm. When Vm is near 0 millivoltage or positive, as occurs, you have seen, during the plateau phase, that is phase 2, little or no potassium current flows. The substantial GK that prevails during phase 4 of cardiac action potential is largely due to the IK1 channels. But currents through these channels is greatly diminished during the plateau. So activation of different types of potassium channel varies according to the different voltages. Other potassium channels play a role in phase 2 of the action potential. These are characterized as delayed rectifier channels. Delayed rectifier channels. These channels are closed during phase 4 and are activated very slowly by the potentials that prevail towards the end of phase 0. So these channels, you should remember that these channels, these delayed rectifier potassium channels are closed during phase 4 and they are activated slowly during the end of the phase 0, that is the early repolarization. When it ends, these channels, they become active. So activation of these channels increases the potassium conductance very gradually during phase 2. These channels plays a minor role during phase 2, but they contribute to the process of the final repolarization, that is phase 3. Two types of IK channel exist depending on the rates of activation. The more slowly activating channel is known as IKS channel, slowly activating. So IKS channel. And the rapidly activating channel is known as IKR channel. So two types of channel. One is IKS, which is slow. Another is IKR, which is fast. The duration of the action potential in myocytes in various regions of the ventricular myocardium is determined in part by the relative distributions of these IKR and IKS. So you have seen the different types of action potentials in this picture. So these also depends on the distribution of these two types of channels. In the Plateau, we have told that the calcium influx and the potassium efflux, they balance one another. As a result, we can see there is Plateau. Now we come to the final phase of repolarization, that is phase 3, the phase 3. The process of final repolarization, that is phase 3, where does it start? It starts at the end of the phase 2. So this starts at the end of the phase 2. In this phase, what is happening? Efflux of potassium from cardiac muscle cells, they begin to exceed the influx of calcium. So potassium efflux is more as compared to influx of calcium. At least 3 outward potassium currents, that is ITO, IK and IK1, contribute to this final repolarization phase, that is phase 3. So this phase 3, in this phase 3, you see here in the phase 3, this is contributed at least by 3 types of potassium channels. namely the ITO, IK and IK1. The transient outward ITO, that is, and the delayed rectifier IKR and IKS currents help to initiate the repolarization. The duration of the plateau is subsequently less in atrial than in ventricular myocyte because the magnitude of ITO during the plateau is greater in atrial than in ventricular myocytes. You see this picture? What happens? The duration of the plateau is less in atrial than in ventricular myocytes because the magnitude of ITO during the plateau is greater in atrial than in ventricular myocytes. The duration of the action potential in ventricular myocyte varies considerably with the location of these myocytes in the ventricular walls. The ITO and delayed rectifier, that is IK currents, mainly account for these differences. The endocardial myocytes, in which the duration of action potential is least. The endocardial myocyte, you can see in the picture, it is the least. The magnitude of IK is greatest. The converse applies to the mid-myocardial myocytes. The magnitude of IK and the duration of action potential are intermediate for epicardial myocyte. Now we come to the inward rectified potassium current. This does not participate in the initiation of repolarization because the conductance of this channel is very small in this range of membrane voltage. However, the IK1 channels contribute substantially to the rate of repolarization once the phase 3 has been initiated. So initial part, they do not contribute much. But once the repolarization phase of 3 has been initiated, they contribute. Now, restoration of ionic concentration, that is the phase 4. The steady inward leak of sodium that enters the cell rapidly during phase 0 and more slowly throughout the cardiac cell cycle would gradually depolarize the resting membrane voltage if there was no sodium-potassium ATPase pump. Similarly, most of the excess calcium that has entered the cell, mainly during phase 2, are eliminated by the sodium-calcium exchanger, which is a form of secondary active transport. This is also an antiporter. 3 sodium in exchange of 1 calcium. In this picture, we can see the T-type and the L-type of currents. So T-type and the L-type of calcium channels and the currents and the voltages in which they are more active. T-type at lower voltages or more negative voltages, while L-type at less negative voltages. T transient remains open for short intervals, L-type long, so they remain open for longer intervals. We had discussed last day that there is a drug called digitalis, which is used in heart failure. What does this drug do? This drug blocks the sodium potassium ATPase pump. So sodium calcium exchanger cannot work now. As a result, calcium gets accumulated in the cardiac muscle cell, and this increases the force of contraction of the failing heart. Digitalis is used in heart failure. Now let us discuss about the slow response action potential. The slow response action potential. So there are different phases. We have already seen the first response action potential, phase 0, phase 1, phase 2, phase 3. Now let us see about the slow response action potential. Here, the upstroke, you can see here there is no phase 1, and the upstroke is much slower, because this is due to potassium current, not due to the sodium channels. This is mainly due to the calcium channels. This is mainly due to the calcium channels. Opening of calcium channels results in the depolarization. So there is slow rise in the membrane voltage, and the repolarization is mainly due to the potassium currents. It is mainly due to the potassium currents. We have already discussed about the pre-potential, where there is opening of a channel which causes influx of, these are known as 8 channels, which are activated during mainly the pre-potentials, which are activated mainly during the pre-potentials or the pacemaker potentials, and they are also known as F channels or funny channels, because they behave in a peculiar way. They behave in a peculiar way. The conductance of this IH increases when the membrane is somewhat hyperpolarized. The pacemaker potentials, rhythmically discharging cells, have a membrane potential that after each impulse declines to firing level. Thus, this is called the prepotential or the pacemaker potential. The prepotential or the pacemaker potential. In the cardiac muscle, depolarization, there is, you can see here, depolarized part, repolarized part. There is rapid transmission of impulses, because the cardiac muscle, they act as a functional syncytium, because there are gap junctions. There are gap junctions. The funny channels, they are activated following hyperpolarization. As the IH increases, the membrane begins to depolarize, forming the fast part of the prepotential. Calcium channels open. We have already told calcium channels are of 2 types, the L type and the T type. There are 2 types of calcium channels. One is the L type, another is the T type. There are 2 types of calcium channels. The calcium channel opening, that is the T type calcium channels, completes the prepotential and Finally, there is gradual rise of the membrane voltage. Next is due to the opening of the L type of calcium channel. Other ion channels are also involved, and there is evidence that the local calcium increase is from the release of calcium from sarcoplasmic reticulum. That is the calcium sparks. This also occurs during the pre-potential. So the action potential in the AC and the AV nodes are largely due to calcium and not due to contribution from the sodium influx. So this is the difference between the conducting tissue and the pacemaker cells of the cardiac muscle. Here you can see the atria, the ventricle, the conducting system, the AC node, the AV node in the cardiac muscle. The pacemaker potential, the slow response action potential of the cardiac muscle, which is generated by mainly due to the influx of calcium ions. And the pre-potential, you can see here the pre-potential. What is pre-potential? This is the rhythmically discharging cells have a membrane potential that after each impulse declines to the firing level. And this pre-potential or pacemaker potential triggers the next impulse. This triggers the next impulse. So let us revise about the excitation contraction coupling in the cardiac muscle. What happens when an impulse comes or an action potential travels down the sarcolemmal membrane along the T-tubule? You can see here there are voltage gated calcium channels, dihydropyridine receptor channels, which open due to the change in the membrane voltage. As a result, there is calcium influx into the cell. This calcium causes opening of the ryanodine receptor channel that is present in the sarcoplasmic reticulum. You can see in this picture the voltage gated dihydropyridine receptor channel. They have opened and there is influx of calcium, which is now acting on the ryanodine receptor channel, And this is chemosensitive channel. In the skeletal muscle, these were mechanosensitive channel. In the cardiac muscle, they are chemosensitive channel. So they open. And as a result, calcium is poured from the sarcoplasmic reticulum into the sarcoplasm. This is called calcium-induced calcium release. And this type of coupling is known as electrochemical coupling. While in the skeletal muscle, it was electromechanical coupling. This is because the isoform of the ryanodine receptor channel in the cardiac muscle and in the skeletal muscle are different. The isoform are different of the ryanodine receptor channels. An abnormality, genetic abnormality of this ryanodine receptor channel can result in malignant hyperthermia. So there is pouring of calcium from the sarcoplasmic reticulum to the sarcoplasm. This calcium binds with the troponin C. 4 calcium binds with troponin C, and the troponin-tropomyosin complex moves deeper into the groove of the actin filament. We have discussed that in the skeletal muscle and the cardiac muscle, the excitation contraction coupling, it is thin filament regulated, because the active site of the actin filament needs to be uncovered for attachment of the myosin head with the active site of the actin filament. While in the smooth muscle, it is thick filament regulated. The myosin head doesn't have ATPase activity out there. Here, there is ATPase activity of the myosin head, and the myosin head breaks down ATP into ADP and PI and stores it like a corkscrew. So when the active sites of the actin filament are free, the myosin head goes and attaches with the active site of the actin filament. And this causes the power. Finally, whenever it attaches, there is then power stroke, tilting of the myosin head, same like the skeletal muscle. And once there is release of ATP, and there is attachment of again another ATP. Once ATP attaches with the myosin head, it loses its affinity for the active site of the actin filament. So what happens? It again breaks down ATP into ADP and PI and gets attached to the next active site of the actin filament. In this way, the myosin head walks along the actin filament, pulling the active sites of the actin filament along with it. This causes decrease in length of the sarcomere and shortening of the muscle. So this is known as sliding filament theory, walk along theory, or the ratchet theory. So when the greater the initial length of the muscle fiber, more will be the force of contraction within physiological limit. That is the Frank-Stirling's law. That is the Frank-Stirling's law. Now, this calcium is the sarcoplasmic reticulum has released calcium. So there is more calcium now inside the sarcoplasm. The muscle will continue to contract until this calcium is pumped by the sarca pump back into the sarcoplasm, or by the sodium calcium exchanger, which is a form of secondary active transport out of the cell. 3 sodium in exchange of 1 calcium. The energy gradient and the electrical gradient is derived from the running of the sodium potassium ATPase pump. Now, what will happen if there is beta adrenergic stimulation? The sympathetic nervous system is stimulated when we get excited. So there is a fight and flight response. In this case, the heart, what happens? So there is release of epinephrine and norepinephrine. Epinephrine we know from the adrenal medulla, and norepinephrine from the sympathetic fibers. So they activate beta adrenergic receptors on the cardiac muscle cells, which in turn activates adenine cyclase and increase CAMP, and thus promotes CAMP-dependent phosphorylation of different proteins in cardiac muscle cells. Both voltage-gated L-type of calcium channels responsible for the trigger calcium and a protein associated with the sarca pump called the phospholumban are phosphorylated by calcium-dependent protein kinase. So they are phosphorylated. This is the phospholumban. So this keeps the sarca pump somewhat inhibited. This keeps the sarca pump somewhat inhibited. So this has been phosphorylated. So what will happen? this will no longer now inhibit the Sarka pump. So the Sarka pump is able to pump back calcium into the sarcoplasmic reticulum. So less calcium moves out of the cell via the sodium calcium exchanger. So during the next period of contraction, more calcium, the next phase period of contraction, more calcium is available in the sarcoplasm as well as inside the cell for making the contraction more effective. So there is increase in force of contraction, that is called positive inotropic effect. There is also an increase in the rate of relaxation accompanying this beta adrenergic stimulation that results in shorter contraction. This increase in the rate of muscle relaxation is termed as positive lucitropic effect. The frequency of contraction of the heart also increases with beta adrenergic stimulation, and this is termed as positive chronotropic effect. So the beta adrenergic stimulation, what does it do? It stimulates the heart, produces stronger, briefer, and frequent contractions. So increase in the force of contraction is positive inotropic effect. Increase in the rate of muscle relaxation is positive lucitropic effect. The frequency of contractions of heart also increases with beta adrenergic stimulation, and this is called as positive chronotropic effect. So next day we will discuss about the smooth muscle, the action potential, mechanism of contraction of the smooth muscle. We will discuss about the calcium calmodulin complex, the myosin light chain kinase. We will see there are some differences between the structures of the smooth muscle and the cardiac and the skeletal muscle. We will see Z lines are absent. Instead of that, dense bodies are present. So we will discuss about them in the next class. Go back and read about the action potential of the skeletal and the cardiac muscle. Bye. See you.